All life is built from proteins, folded into skins, seeds, silk, feathers, and muscle. They form the quiet fabric of bodies, harvests, meals, earth, and microbial worlds. In every cell, their long chains curl into shapes with precise, particular work. But proteins are not loose threads, waiting politely to come apart. Their links are strong, repeated, and hidden inside compact, beautiful, living architecture. For nourishment, renewal, and industry, those shapes must often be opened safely. Water alone does little. Time alone usually moves too slowly for human needs. Acid adds pressure, but even acid needs a careful helper inside the meal. That helper is acid protease, a wet cutter made for sour places. Long before laboratories named it, stomachs already knew its ancient patient craft. A swallow enters darkness, where acid and enzyme immediately begin working together. Meat softens, milk curds loosen, and proteins become small usable fragments. The same solution appears in fungi, feeding quietly on fallen fruit and forest leaves. Molds release acid proteases into damp matter, harvesting nitrogen from decay again. Nature had solved the locked protein problem without flame, force, metal blades, or boiling vats. Human observers first met this mystery by wondering about the stomach inside us. In the early 19th century, digestion was still partly mechanical imagination. Some believed food was crushed, warmed, and dissolved by simple passive juices. Then came William Beaumont and an accident on the young American frontier. Through Alexis St.'s healed wound, Beaumont watched digestion directly. Food disappeared in gastric juice, even outside the body in a vial, without chewing. The stomach was not merely grinding, something invisible chemical was acting there. In 1836, Theodor Schwann carried the clue into a working laboratory. From animal stomach lining, he prepared an active digestive substance carefully. He called it Pepsin, from the old Greek word meaning digestion. It worked best in acid, and it attacked proteins with its unusual specificity. Pepsin became one of the earliest enzymes recognized by modern scientific history. It gave acid protease its first famous face and a nameable human story. The discovery changed digestion from hidden bodily warmth into measurable experimental chemistry. Decades later, John Northrop patiently coaxed pepsin into crystals in a laboratory. Those crystals helped prove that enzymes themselves could be living proteins. The cutter of proteins was also, beautifully, a protein. Physicians and pharmacists soon valued pepsin as a daily practical extract. 19th-century apothecaries handled stomach preparations with glass funnels, ceramic jars, corks, and bottles. Industry's first commercial acid protease story began with animal tissue. But animals were limited sources, tied to slaughterhouses, seasons, costs, and uneven supply. A broader answer was already growing on warm steamed rice and soybeans. In East Asian fermentation, koji molds had long transformed proteins gently and patiently. Their enzymes made deep, rich flavors from grain, beans, salt, and thyme. In the 1890s, Jōkichi Takamine brought koji fermentation into modern industrial science. His work showed microbes could be cultivated as dependable enzyme partners for useful work. Among their many gifts were proteases, suited to acidic fermenting foods. During the 20th century, fungal acid proteases became important industrial tools by mid-century. Species of Aspergillus and Rhizopus offered hardy enzymes beyond the animal stomach. Fermentation turned tiny fungal meals into useful protein-cutting preparations without needing animals. The story had moved from organ extract to renewable microbial abundance. At the microscopic scale, acid protease is neither sharp blade nor tooth. It is a small folded molecule, holding a pocket shaped by evolution. A protein strand drifts close like seaweed and settles into that pocket. In sour water, the enzyme precisely positions one chosen link. Then, almost quietly, it encourages water to split the bond cleanly. The enzyme is left unchanged, ready to welcome another strand. No fire is needed, and no violent harsh pressure commands the change. It simply lowers the barrier nature patiently placed around the reaction. The results are smaller peptides, softer textures, and more accessible nutrients. In cheesemaking, acid proteases help milk proteins find new form during ripening. In fermented foods, they slowly release savory depth from beans and grains. In brewing, they can quietly help manage proteins during complex fermentations. In baking, they may relax delicate dough and improve gentle handling. In animal nutrition, they help unlock varied feed proteins more efficiently. In leather processing, enzymes can soften hides with less traditional aggressive chemistry. In protein hydrolysates, they create balanced fragments useful for foods and bioproducts. In laboratories, Pepsin still helps scientists study complex antibodies and tissues. Every use rests on the same quiet molecular preference for acidity. Where many enzymes fail, acid protease still keeps working in sour surroundings. That resilience makes it valuable in wet material streams once considered difficult. It can sometimes replace heat, shorten wasteful steps, and spare stronger chemicals. The old stomach mystery has become a tool for gentler, cleaner making. We learned carefully by watching bodies, then by listening to microbes. In a greener world, patient sour enzymes may keep opening what once seemed locked. At Enzymes Bio, we're a small team of scientists and biotech enthusiasts. We bring the world's enzymes onto one platform, so startups and labs skip the procurement maze. A greener chemistry for the years ahead. Learn more at Enzymes Bio.